Ivy League institutions. They are two of the country's best schools. She was appointed to a lifetime position on the Supreme Court by President Barack Obama in 2009. Of course, he was the 44th president. And in the history of our country, she was the first Latina woman and only the third woman in general confirmed to the Supreme Court of the United States. So her confirmation was a big deal. It was historic. She has been a very important member of the Supreme Court in her time on the Supreme Court. And getting to the Supreme Court is a big deal because it's a lifetime appointment, meaning that there are only really three reasons or three ways that you could no longer be. Once you've been appointed, there are only three ways that you could no longer be on the Supreme Court. Number one is death. Number two is retirement, if you choose to step away and you want to spend more time with your family. And then the third way is by being impeached, which has never happened in the history of the country. So getting to this point in your legal career is a big deal. And I wanted to take my quote from her because she is such a historic figure. The quote is, "I have never had to face anything that could overwhelm the native optimism and stubborn perseverance I was blessed with." So I want you all to think about that quote as you head into the end of this quarter and you start to think about the start of the second quarter. You do have a 3-day weekend this coming weekend because we don't have school on Monday as it is a teacher work day. So I want you all to get yourself in the mindset to head into the second quarter ready to work hard, ready to take some of the successes that you had in the first quarter and build upon them. I know some of you do not have the glass half full rosy optimism that some people are born with, as Judge Sotomayor spoke to, but you can still think about having some momentum. So even though you may not be exactly where you wanted to be at this point in this semester, you still have plenty of time. You can still look back and say, you did this well, you did this well. We've all done a lot to persevere in this time of pandemic. We've all done a lot to persevere in this time of virtual learning. We've had to adapt. We've had to communicate. We've had to be resilient. We've had to be understanding. We've had to be forgiving. So for all of those reasons, you deserve to look at yourself in the mirror and say, I've done well, but I can do better. I can continue to persevere over these struggles. All right, so keep that in mind as we head into this week. One more thing before we jump into the biology lesson. Today is Monday, October 12th, meaning that is a celebration of what we have historically called Columbus Day in recognition of Christopher Columbus, who in 1492 landed on what we today think was the coast of Jamaica. And he thought he had landed in India, and he was in search of India. He had left from Spain. He was a Portuguese man, but he had been funded by the Spanish royal family to travel to look for a sea route from Europe to India. India at the time was producing huge amounts of spices. So the spice trade was extremely lucrative. It was extremely economically beneficial to the Europeans. And so they were looking for a way to get the spices from India to Europe without having to cross vast amounts of land. So Christopher Columbus sails thinking that he'll eventually reach India, but what he actually lands on is the Americas, what we know today as the Americas. So some people have said that this was a discovery of the Americas, that he discovered this new world and opened up the world to all of the opportunities and all of the resources that were present in this new world. But over the last 29 years, various cities in the United States have celebrated, instead of Columbus Day, Indigenous Peoples Day. And this is to recognize the fact that Native American people were here before the quote unquote discovery of America by Christopher Columbus. And there were many, many thousands and by some estimates, millions of Native American people who were living in the Americas. And of course, their population was decimated when the Europeans arrived and brought with them a mentality of war, brought with them a mentality of genocide, and they brought with them many diseases as well. But we have to choose to celebrate both sides of the history. instead of just recognizing the quote unquote discovery of the quote unquote new world by Christopher Columbus, we also have to acknowledge that before that discovery, there were also people who were here. And in 1977, during an international conference in Geneva, Switzerland, attendees decided to, quote, observe October 12th, the day of so-called discovery of America, as an international day of solidarity with the indigenous peoples of the Americas. And the indigenous peoples they are referring to are, of course, the Native Americans, the people who, for thousands of years and many hundreds of generations, were living here in what is now known as the United States and Canada and Mexico and Colombia and Argentina and Paraguay and Uruguay and Peru. All of those people were here before the European people came and colonized it. Yes, the European people brought with them a specific culture, and the cultures of the European people intermingled with the cultures of the indigenous people, and then eventually intermingled with the cultures of the African people who were brought here as slaves. But we have to choose to recognize the history of the indigenous people, primarily because for so long that history has been ignored or it has been overlooked. All right, so I want you all to think about Indigenous Peoples Day today. You may have heard of Columbus Day, maybe you've heard of Indigenous Peoples Day, but it's an opportunity for us to think about the many contributions of Native American people to our culture, and many of their contributions to our economy, many of their contributions to our scholarship, and all the things that make this country what it is and make the Americas what they are. All right. That being said, let's go ahead and start thinking about biology. So at this point, you should be taking out your notebooks. And you should be writing down the title of the lesson, which is Cell Specialization. As well as the date, which is October 12th, 2020, and Unit 5, Day 5. And as you all finish writing that up, I want to read the objective to you. The objective states that biologists will be able to explain how instructions in DNA lead to cell differentiation, meaning specialized cells that perform specific functions. The essential question, how do the cells in multicellular organisms perform so many diverse functions? How do the cells in multicellular organisms perform so many diverse functions? Now, I am going to ask you that question again at the end of today's lesson. So I want you to, as you take notes, be intentional about how you're taking your notes. Words have been highlighted, and I want you to write down those highlighted words, but I also want you to try to answer this essential question at the end of the lesson. So maybe some of you might find it beneficial to write down that essential question. And Jaden, I see your question in the chat. Yes, we are taking notes. All right. So quickly, I want to review the general process of protein synthesis. Of course, protein synthesis is the process of synthesizing or making proteins. That process requires two first steps. Number one, transcription. Transcription is the process by which DNA is transferred into an mRNA strand. We call this transcription because it is the same language. It's the language of nitrogen bases. So going from DNA to mRNA, messenger RNA, is in the same language, and we call that transcription. The next step is translation. And this is when we go from mRNA to amino acids. mRNA, of course, is it's a nucleic acid, so it's written in the language of nitrogen bases. Amino acids are written in a language that is more like proteins. That is the beginning of a protein. So we have to translate those languages. This is why we call it translation. So we see that transcription comes first, and it is followed by translation. This is the central dogma of biology, meaning it is probably the most important principle of the study of life. And that principle says that genes code for the proteins we make. They are the instructions of the proteins that we make. A cell's primary goal is to make proteins. So the cell relies on its genes to make those proteins. Okay. So I want you guys to write down what you see highlighted in your preferred language. Yellow for English, green for Spanish. I apologize if your preferred language is not one of those two. Please contact me if you'd like me to translate the slides into another language. But a gene is defined as a small section of DNA that codes for a specific protein. A gene is just a small section of DNA that codes for a specific protein. So, for example, human DNA has approximately 30,000 genes. So we have this long strand of DNA, and along that strand are 30,000 smaller sections that we call genes. Each of those genes is responsible for making an average of 3 proteins. So there are a lot of genes in the human body. You've heard of some of these proteins. We've talked about some of these proteins in class: hormones, insulin, hemoglobin. What else did we talk about? Enzymes, and what's the last one? Antibodies. So we talked about those 5 proteins, but there are many others that you will never have even heard of, but they're still responsible for carrying out important functions for the cell. Okay. So would someone like to read that sentence at the top of this slide?
I'll read it for you.
Thank you.
Gene regulation refers to the process of activating, turning on, and deactivating, turning off certain genes.
Thank you so much. So gene regulation is basically like a light switch. That's how we can think of it. So we've got those 30,000 genes all throughout our DNA, but not all 30,000 of them are turned on at once. We can think about this as if you lived in a mansion with a lot of different rooms in it. At any given time, you're not going to have the lights on in every single room. Our cells are the same way. At any given time in that mansion, You wouldn't want to waste energy and have to pay a light bill for rooms that you're never even in. So you keep the lights off. Our cells don't want to waste energy making proteins that they don't need. So they turn those genes that code for those proteins that they don't need, they turn those genes off. They don't need those genes. But the genes that they do need are turned on, just like in your mansion. Maybe you only step foot in 4 rooms: the master bedroom, the kitchen, the bathroom, and the living room. So you're always in those 4 rooms and you're just turning the lights on in those 4 rooms. Everywhere else, bedroom number 2, 4, 5, 6, 7, blah, blah, blah. You don't need those lights on in those extra bedrooms. The basement, you don't need the lights on in there. The second kitchen, you don't need the lights on in there, depending on what your mansion is looking like. You only turn the lights on that you need. And our genes, our cells are the same way. They will only turn on, they will only activate the cells that they need. I'm sorry, they will only activate the genes that they need. All right. But it's important as we think about this process that you all realize that all cells have an entire copy of the DNA. All cells have the entire copy of DNA. So it's not that some cells have this portion of the DNA and the other cells have that portion of the DNA, and that cell over there has a third of the DNA. Every cell in your body has your entire genome in it. It has your entire DNA. The difference is that some cells have specific genes turned on and others have other genes turned on. Okay, so think about this mansion. Let's say that you have a mansion and your best friend's mansion is next to you. Your best friend doesn't ever cook, but they spend a lot of time in their game room. You like to cook a lot, but you don't even have a game room. Well, you have a game room, but you don't like to use it. So your lights are always on in the kitchen, but never on in the game room. Their lights are never on in the kitchen, but always on in the game room. Okay, so our cells are the same way. They can decide based on what their needs are, which lights to turn on and which to turn off. Again, this is all about the production of proteins. This is all about protein synthesis. The goal of any cell is to produce a protein or several proteins. And gene regulation is how cells control which proteins they produce. Okay, so in this image in the middle of your screen, we can see that there's this long strand of DNA. And along that strand of DNA, we've got 1, 2, 3, 4 genes. Gene 2 and gene 3 are switched on. So they are producing a protein. They're producing 2 different proteins. Gene 1 and gene 4 are off. So they're not producing any proteins. So this whole process is about determining which proteins get produced and which proteins don't. Not every cell needs to make every single protein. Each cell is responsible for doing a very specific job, and so therefore it needs to make specific proteins. So in order to do that, a small portion of the genes will be turned on. Everything else is turned off. It's not needed. We don't want to waste energy doing it. So we turn the genes on when we want to produce a protein and off when we do not want to produce a protein. A cell knows which genes to turn on and which genes to turn off because cells are always communicating with one another. So gene regulation, the process of activating genes or deactivating genes, is controlled by electrical signals and by chemical signals. Electrical signals are carried by neurons. Those are your nerve cells. They carry an electrical charge. Your chemical signals are carried by hormones. And your cells are always sending these messages to one another, telling them what to do and when to do it and how much of it to do. When should I produce this protein? Where should I produce this protein? And how much of this protein should be produced? So gene regulation is controlled by this cellular communication. The cells are always communicating with one another. One thing that we'll talk more about later in the course, but one thing that I'd like to mention right now is that there's a constant interplay between our genes and our environments. And both have an impact on the proteins that our cells produce. So our genes interact with our diet, the activities that we frequently do, as well as any exposure we might have to chemicals or radiation. And our genes are affected by those things. So in this case, we have two identical twins. They both have the same gene that absorbs fat twice as fast as the average person. So they both are more likely to gain weight because their body absorbs fat more readily. But one twin chooses to eat healthily and they eat food that's low in fat. And so the genes that she has that make her more likely to become obese are never actually triggered. The other twin eats an unhealthy diet that is consistent of many fattier foods. And so those obesity genes, as this image calls them, are triggered. So there's this interplay. Your genes don't totally determine what will happen to you, just like your environment doesn't totally determine what happens to you. They both have an impact in making you who you are. They both have an impact in the proteins that your cells produce. And the results of those proteins are traits. Just like a person with curly hair can get their hair straighter by putting chemicals in it or by applying heat to it. And a person with naturally straighter hair can curl their hair up by putting different chemicals in it. Your genes are influenced by your environment. Okay. So October is Breast Cancer Awareness Month. Many of you are aware of that, I'm sure. And you see people wearing pink during the month of October. Can anybody tell me how a person's likelihood of developing breast cancer is affected by both their genes and their environment? How does a person's likelihood of developing breast cancer get affected by their genes and their environment? How is a person's likelihood of developing breast cancer affected by both their genes and their environment? Okay, so maybe we don't have any ideas about that, but that's okay because that's what I'm about to talk about. So there are two genes on two different chromosomes in the human DNA that have been shown to impact a person's likelihood of developing breast cancer. They're called Broca genes, BRCA genes, stands for breast cancer genes. These Broca genes are found on two different chromosomes, chromosome 17 and chromosome 13. Now, we all have those genes, but again, gene regulation determines whether or not the genes are turned on. And then we also know that those genes can experience mutations. So a mutation in the BRCA1 gene, which is on chromosome 17, greatly increases the likelihood of developing breast cancer by age 70 to about as high as 65%. The mutated BRCA2 gene on chromosome 13 also greatly increases the likelihood of developing breast cancer by age 70 to about 45%. So a person who has normal BRCA genes, where there are no mutations, has only about a 12% chance of developing breast cancer by age 70. All right, so this is very important. This really allows people to proactively look out for breast cancer so that it can be caught early and treated aggressively before it spreads and becomes a much greater health issue. And some women are actually starting to remove their breast tissue before breast cancer can even develop, because they know that they have a high likelihood of developing breast cancer. Okay, so these genes, mutations in these genes, greatly increase the likelihood of developing breast cancer. But even though we know that there's a genetic side, we also have to acknowledge that there are other factors, including some factors that are controllable, that determine or at least increase the likelihood of a person developing breast cancer. So a woman who chooses to have children late, so let's say a woman who has children after the age of 35, is more likely to develop breast cancer. It's not a guarantee. It's not to say that that will definitely happen. It just means that the risk is greater. Women who choose to consume alcohol or who smoke tobacco are also at a greater risk of developing breast cancer and many other types of breast cancer. And then there are some other factors that are not up to choice, like the starting age of menstruation or the starting age of menopause. Neither of those is really under a woman's or a female person's control, but they can affect when a person develops breast cancer, or the likelihood that that person will develop breast cancer. Okay. So back to writing. You guys didn't need to write anything on those slides, but again, the words that are highlighted in yellow should be written down. And I apologize for not translating this slide into Spanish. So gene regulation results in cell specialization. Gene regulation results in cell specialization. So genes are turned on or turned off, and it leads to them having special jobs. Cell specialization is the process by which cells produce specific proteins and therefore develop different structures, which results in different jobs and abilities. We also have to remember that structure determines function. So as a cell changes shapes, it's going to change jobs. It's going to have different abilities. So as it produces different proteins that change its shape, it's also going to have different abilities based on that shape. Structure determines function. Okay. So stem cells are undifferentiated. So we just talked about the process of cell differentiation or cell specialization. They mean the same thing. They are undifferentiated, meaning that they have the ability to develop into any number of different cells. So they are what we call undifferentiated. They haven't been assigned a job yet. So they have a lot of potential to become any number of things. Now, as we look at this image in the middle of your screen, what process are we seeing? What process is depicted here? Can anyone tell me that? What process are we seeing in that image? Okay, Nick says interphase. You're close, Nick. What comes after interphase? What comes after interphase? And Jaden, no, you all do not have school next Monday. What comes after interphase, folks? Anyone can answer. It doesn't just have to be Nick.
Meta.
Okay, so I heard Brian say meta.
G1 phrase.
Jaden says G1. So G1 is the first part of interphase. So we have G1, we have synthesis, and then we have G2. Those are all a part of interphase. But what comes after that? What's the process of cell division? Thank you, Kyrie. Yes, we've got mitosis. So mitosis is the actual process of cell division. And we can see that taking place here. We start off with one cell. That cell divides and becomes two cells. That cell divides and becomes four cells. That cell, those four cells become 8 cells eventually, and we keep dividing. All right, so what we're seeing here is mitosis. So stem cells undergo frequent mitosis, and they are undifferentiated, which gives them a lot of, and I see you said it too, Nick. I'm sorry, I didn't see your answer at first. Mitosis is the right answer. So we have a lot of potential with these stem cells because they have the opportunity to become any of these other types of cells. A stem cell could eventually develop and become differentiated. It could be a muscle cell. It could be differentiated and become an immune cell. It could become differentiated and become an epithelial cell, which is in your skin. So as their genes are regulated, as I turn on some genes and turn off other genes, I start to develop different shapes and different functions that give me different abilities. So cells start out as undifferentiated. And so what we're seeing here is how cells start out. And eventually they develop and they start to get different jobs. And they're communicating with each other all the time. So in the earliest days of development, between fertilization and embryonic development, the growing human cells are primarily stem cells. They start off as undifferentiated, but they are destined to become specialized based on a process of cellular communication. So the cells will say, hey, that group of cells is located where we want the head to eventually be. So they need to start developing into brain cells and ears, cells that are in your eardrum, and cells that are in your eye, and cells that are in your taste buds. And those cells are located eventually where we want the back to eventually be. So they need to start to develop into spinal cord cells. And eventually you end up developing a baby that, or a human being that is made up of many thousands of different specialized cells. But the cells start out as undifferentiated. They start out as stem cells. So in summary, I want to revisit the essential question of the lesson, and one of you should unmute yourselves and try to answer this for me. The question is, how do the cells in multicellular organisms perform so many diverse functions? How do the cells in multicellular organisms perform so many diverse functions? Can someone try to answer that question for us? Kyrie says different tissues are in turn grouped together to form larger functional union. Okay, I don't know where you got that answer from. Yeah, okay. It sounds like a Google answer. But based on what we just talked about, guys, how do some cells do one job and other cells do another job? What is actually controlling the jobs that they do? Cell specialization is a part of it, but what results in cell specialization? What results in cell specialization? What causes cell specialization? I want to stick with what Nick said, because we didn't mention tissues at all in this lesson. How do the cells in multicellular organisms perform so many diverse functions? How do they do so many different jobs? It's all about cell specialization, which is a result of gene regulation. Some cells have certain genes turned on and others turned off. Other cells have different genes turned on and others turned off. So the types of genes and the number of genes that are turned on determines the job that the cell is able to do. It determines the function of the cell. All right, so we've got to think about this as a matter of gene regulation. I think that the exit ticket and the asynchronous assignment will help you all better understand that. They're both short. We have 20 minutes left, and that should be plenty of time to knock them both out. We got 21 minutes. So I'll be here on mute, but I want you all to go ahead and knock out those 2 assignments to fully understand what the goal of this lesson was.
Hey, Mr. Rudd, did you get my email?
Who was that talking just now?
Sean.
Okay. Yes, I got it. I think so.
Uh,
Let me see. Yeah, I got it. I'll get back to you shortly.
all right, bet.
Okay, folks, sorry, I lost track of time a little bit. I was taking care of some work, but it's 1:22, so you all are good to know, good to go. And I hope you enjoy the rest of your Monday. Please let me know if you need anything.